So welcome everyone to, I think, is this the final seminar in this seminar series from the IICD? We're very grateful today to have Samra Turajlic with us, not only because she's going to tell us about some very exciting research, but also because it's quite late in the UK already. So we're additionally grateful that she's attending. So just an introduction to Samra. She completed her undergraduate studies in the University of Oxford and clinical training at UCL Medical School, and then moved on to get a PhD in 2013 from the Institute of Cancer Research. In 2014, she was awarded a CR UK Clinician and Scientist Fellowship to study cancer evolution at the Crick Institute, and completed her training in medical oncology. After that, she was awarded a consultant position at the Royal Marsden and became an independent group leader at the Francis Crick Institute in 2019, where she is currently dividing her time between the clinic and her lab. So I think I'm particularly fascinated by Samra's research because she really adds the dynamics to cancer evolution, and that's mainly, well, in part at least, through her initiative, TraceX, on renal cell carcinoma, where they really have done incredible stuff collecting data on a huge cohort of patients. And I think that's an ongoing initiative that she leads. But among other things, she's also worked on other cancer types. She also works on melanoma, I think. And she's here today to talk about deciphering cancer evolutionary dynamics. And we're very happy to have her here. So I think with that, I'll hand you over to present Samra. Thank you very much.
Thank you so much, David, and thank you for the kind introduction. It's a real pleasure to have been invited to speak to you all. And you know, I would love to visit the Institute at some point in the future. So here are my disclosures, just briefly. And as David mentioned, I'm an oncologist, so the work that we do in the lab is very much motivated by what we observe in the clinic. And arguably one of the most bewildering observations in the clinic is the variety of outcomes that patients end up having following their diagnosis of cancer, even when they're seemingly similar cancer phenotypes. And of course, the most difficult convergence that we're seeing here is that onto metastasis and treatment resistance, but not forgetting that, you know, of the patients that we treat and believe to be cured, there's probably also a proportion of patients that are undertreated. And certainly in the developed world, this is an increasing issue as well. So we take a cancer evolution approach to this, and it's very much an interdisciplinary approach. The reason why we always start with patient data is because this is our ground truth. And as I'll go to show you in a moment, the sort of population size and the temporal dynamics of cancer in patients is very difficult to recapitulate in preclinical models. And certainly some of the more extreme evolutionary trajectories and observations that we make in patient tumors would be very difficult to observe without the time and the space that is available in cancer evolution in vivo in patients. However, each instance of cancer in a patient is a unique instance of cancer that can't be repeated. And of course, we'll have some unique features relative to that patient's germline, their environmental exposures, the treatments that they've had, and so on. So that really limits our observations in terms of understanding their repeatability. And this is where, in the recent years, we've started working with mathematical modelers as well. This has been a very enlightening move for our lab, and that I'm very excited about. And this really allows us to run those repeatable experiments and discover features that we wouldn't in a sort of time-resolved fashion that simulations allow for. What we also seek is to refine the parameters that we provide into these models, and that's much easier to do in preclinical models, where we can really dissect out the fitness that's afforded by different genetic alterations, different combinations of genetic alterations. And we're doing this through preclinical models, mostly in organoids or genome editing. And then lastly, you know, we're very mindful of how labor-intensive our approaches are to deriving these evolutionary features in patient tumors that would allow us to predict the cause of cancer, and the huge barrier to adoption of these methods in the clinic. So we're very much focused on developing more tractable methods to get this into the clinic. So this is the structure of the talk. I might not be able to get through all of it, so I might skip through as we go along. I'll keep an eye on the time, but we're not that many, so I would love it if people just interrupt and ask questions as we go along as well. And I'll try not to spend too long on the published data and give you a flavor on what we're working on at the moment. So starting with these with the patient cohorts, this has really been a fundamental cornerstone of what we do, and the way that we sample the ground truth is opportunistic. So when tumors are getting biopsied or surgically resected, when they recurred or become resistant to treatment. And we're really enormously grateful to our patients and their families, who are such important stakeholders in this. And this has been particularly true of our post-mortem studies, where obviously we're able to get to areas of the body that are not easily accessible in life. So there are various outputs from this program, and we have a variety of projects that are derived from this. But really today I want to focus on sort of clonal reconstruction from mutational data and how that relates with the clinical phenotype. And why kidney cancer? Well, clear cell kidney cancer specifically, which is the most common type of kidney cancer that derives from the proximal tubule of the nephron, which is the basic unit of the kidney. It is a beautiful model of cancer evolution. First of all, it provides a wide range of population sizes. So incidentally discovered kidney tumors, which nowadays happen through sort of routine imaging for other reasons, the primary tumor population size might stretch into tens or hundreds of millions of cells. But the more advanced cancers will frequently reach more than 1 trillion cells. And you can immediately imagine how that changes the mutational supply and the variety of tumor microenvironments that might be experienced by these tumors, and how that can shape clonal evolution. Also, there is a wide range of clinical behavior. So, for example, these cancers that are detected at a very early stage, and I talked about overtreatment earlier, studies of active surveillance in these patients show that many of them don't progress at all. They show very limited growth over 10 to 20 years, but there is around 10% that do progress and might metastasize. So this suggests that some of these tumors at least are in some sort of equilibrium, and they're not destined to become larger tumors. And of course, this is well described in some other settings, such as prostate cancer. What's more bewildering still is the types of metastatic manifestations that we get in kidney cancer. So when we see patients who present with these very large tumors, we resect them with curative intent, essentially, and these are the sorts of scenarios that these patients end up with. So some of these patients will, at their first follow-up scan, have a widely disseminated disease, which means that they had occult metastases all along. They just were below the level of detection by CT. Some of them will exhibit this sort of intermediate metastatic efficiency, which is quite peculiar, and it's relatively specific to kidney cancer, although at a high frequency. It does occur in other cancer types too, and it does have some organotropism as well. So these would be solitary metastases without broader metastatic spread. So the question is really what's constraining more comprehensive metastatic spread in these patients. And then there are patients that will go on for another 10 or 20 years, and then they will have this latent relapse, and it's very often in endocrine organs for reasons that are unknown. These patients do extremely well, and these metastases are often isolated. They're believed to represent dormancy, but we don't really know whether that's the case, or at least we didn't. And then probably the most compelling reason why clear cell RCC is a good way to study cancer evolution are these beautifully constrained genetics. So the first event in formation of clear cell RCC is loss of the short arm of chromosome 3. Now this, the region that's lost is variable, but it always contains these 4 tumor suppressor genes, VHL, PPRN1, SETD2, and BAP1. Therefore, just this one event results in the loss of heterozygosity in 4 tumor suppressor genes, and therefore you can probably guess that the subsequent events really all involve inactivation of the remaining allele of these tumor suppressors, and indeed that's what happens. The most common second event is either mutation or methylation of VHL, this is a relatively consistent founder event. Of course, you know that the role of VHL is in hypoxia sensing, and what loss of VHL results in is this state of pseudo-hypoxia. So in normal oxygenation conditions, the hypoxia response is switched on, and this leads to angiogenesis. And really, angiogenesis is a canonical feature of the environment in clear cell RCC. But we know that this biallelic loss of VHL isn't a particularly strong driver of tumor growth, because most tumors, almost all tumors, will have a second event in one of these remaining tumor suppressor genes. And as I'll show in a moment, a lot of what happens in kidney cancer can be explained through some permutation and combination of these events. Now, copy number drivers are also very well defined in kidney cancer, and very helpfully to us, because we often use off-target reads to determine copy number events. These tend to be either whole chromosome or chromosomal arm losses and gains, and they're shown here. So what we've learned so far is that there are 3 distinct modes and tempo of evolution observed in clear cell RCC. And these, of course, match the modes of evolution that have been described as a framework in cancer evolution literature. But I really want to show you how we observe them in clear cell RCC. So first of all, we see tumors that evolve in so-called linear fashion, although inferring linear evolution is invariably difficult, and in the absence of detecting intermediate populations, we don't know that this is truly linear. But what's evident is that these are monoclonal tumors. There's really no clonal complexity at all. They're very low on driver burden. They just have loss of 3p and VHL, and they grow very, very slowly. Now, when we first observed these tumors and these evolutionary features, we were concerned that what we were observing was a lead time bias, that essentially we were catching these tumors in patients before they turned into this or this. But in fact, through multi-regional whole genome sequencing, we were able to determine the most recent common ancestor to presentation time, and frequently this was several decades, suggesting that some other constraints are responsible for lack of evolvability in these tumors. WGII here, by the way, represents the weighted genome instability index, so it's simply the proportion of the genome that's altered by copy number. And as you can see, this goes up as we go through these modes. The second is more classical Darwinian evolution. So there is a polyclonal tumor here with genetically distinct populations of cells. None of these these drivers are sort of equal in terms of the fitness advantages they provide, and therefore the size of the clones is relatively comparable, and there's lots of parallel evolution of these tumors. They also evolve over long periods of time. And then lastly, we see a very different tempo and features in the context of punctuated evolution. So this refers to kind of an evolutionary shortcut where a highly fit clone, an aggressive clone, is fixed very early on in tumor evolution. And this leads to rapid growth of the tumor. These tumors grow to the same size as these polyclonal tumors, but they do so over a much shorter period of time. And what really fascinated us is that these modes of evolution reconciled some of these clinical observations that we're making. So the tumors that are evolving linearly were these small renal masses that show no progression over long periods of time. I mentioned to you earlier about the tumors that have intermediate metastatic competence, only seeding a single site of metastasis, for example, and they associated where primary tumor was characterized by branched evolution. This is the picture we were tending to see in the clinical context. And this unfortunate group of patients with rapid progression, their primary tumors were frequently characterized by this, sort of born to be bad tumor. But what was further fascinating to us is that these modes of evolution were actually underpinned by highly conserved mutational patterns, and I'll expand on this a little bit on the next slide. So we followed all the possible trajectories across the tumors that we'd examined, and we found statistically significant conserved patterns of mutational ordering, but also timing, whether certain mutations in certain genes were happening early versus late, and also mutual co-occurrence or mutual exclusivity at the level of a clone as opposed to at the level of the tumor as a whole. And these rules essentially follow these three trajectories. So from that early inactivation of VHL, some tumors will acquire mutations in PBRM1, which always then preceded further evolution that involved activation of the PI3 kinase pathway, repeat selection of SETD2 mutations, or selection of copy number drivers. That rapid progression and punctuated evolution proceeded down either acquisition of multiple driver events very early on and fixation of multiple driver events on the trunk, or acquisition of BAP1 mutations, which really seem to outcompete any other populations, and we didn't find evidence of fixation of additional events generally in these tumors. We also, especially in large tumors, and this is really important to highlight in terms of population size and what we observe when we have a large population size, we see extensive evidence of parallel evolution. So here are the samples from a tumor. So this is a 2D slice of a tumor taken from the middle of the surgical specimen, and each of the colors represents a distinct mutation in SETD2. So there is a strong drive to select for these mutations, but there is relatively limited expansion. As you can see, they only expand within a single region of the clone, and we're currently trying to understand also why they cluster in certain areas of the tumor. And when we see extensive evidence of parallel evolution with respect to other genes, so in this case, here is this BAP1 clone. So I told you earlier that BAP1 is a very strong driver of tumor growth. So in this case, we were actually able to sample the tumor in Z axes as well. And we can see that this BAP1 clone really drives a very strong clonal expansion in all axes. But this is not penetrating to certain areas of the tumor, which is perfectly possible because of the physical barriers. And we see repeat selection of BAP1 in other areas here in light green and dark green. But when SETD2 is selected, it's never on the background of BAP1. And these are absolutely mutually exclusive. So that mutual exclusivity is really apparent at the level of the clone. But if we describe mutual exclusivity at the level of the tumor, it would not be apparent. And recently, we've started profiling some of these tumors by single cell sequencing, and it seems that there's really ample fuel for ongoing selection in these tumors. And for example, we see at single cell level selection of other well-known driver genes, but again, mutually exclusive with BAP1. So I'll just move on now to a more recent work, which is really trying to add a spatial component to this understanding of clonal evolution. And one of the utilities of having the framework that we have in the way that we sample these tumors is that we can correlate correlate the exact location of a tumor clone to its genetic sequence. And we can ask a range of questions about the relationship between clones, also clone size, so that we can infer clone fitness from that. So how far does a clone spread And we can also ask questions about different areas of the tumor to try and infer where the selective pressure is coming from. And this really then gave motivation to a mathematical modeler that we were working with, who was very keen to develop a model that links the type of tumor growth to the type of evolutionary outcomes that we see. And we were very privileged to work with Zhao Fu, who's currently a senior postdoc at the Crick, soon to progress to running his own group. And Zhao worked on a spatial model where he was interested to explore how limiting the proliferation in the tumor to only the surface versus allowing proliferation to happen throughout the tumor might impact on the final clonal composition of the tumor. And the reason why Zhao was keen to work with us is because he was able to take the data from the primary tumors to assign certain parameters within this model, such as the fitness advantage of particular genotypes. So when Zhao ran these simulations with surface growth versus volume growth, the first thing that he noticed was that there were obvious morphological differences in how these tumors end up, and I'll come back to this in a moment. But furthermore, surface growth resulted in sort of cancer cell fractions that were very variable, and repeat simulations and always result in polyclonal tumors, while volume growth was more akin to those punctuated evolution events that we were seeing. One of the things to mention is that what we're really looking at here when we're reconstructing these phylogenies is a fairly successful clonal expansion. So we're detecting variants that are present in at least 10 million cells. But we've also been able to get it so microdiversity, which probably speaks to future trajectories of evolutionary trajectories of these tumors. And again, Zhao sees that the spatial distribution of this microdiversity is different. So in a surface growth model, it tends to occur more patchy and towards the surface, while it's more evenly distributed in a volume growth model. And again, we were able to make the same observations in the tumor. I'm rushing through this because it's published data, but please stop me if everything's unclear. Anything is unclear. So the other question we asked Zhao, because again, we don't have time results data in our patients, so it's very unusual to monitor cancer growth over long periods of time. We usually diagnose patients, we take these things out. So we asked him whether he was able to rewind some of these simulations, because we were interested to know whether there is any hint of what might happen morphologically in the tumor before there is detectable subclonal diversification. And what he noticed is pointed out here, these budding structures which preceded detectable subclonal outgrowth and diversification. And then we reached out to our collaborators at the Royal Marsden and the Institute of Cancer Research, who have a radiomic pipeline, and we were able to analyze the radiological images from our kidney cancer study. And in these early stage kidney cancers, we found some evidence of these budding structures. They're pointed out here. So this budding structure on this radiological image actually relates to this clonal map that you're seeing here. And the red color indicates the most recent clones and the number of subclones. So these are the clones that are being born essentially. And so indeed we see subclonal diversification that is happening in these structures. So we were quite excited to see this, because again, we're always mindful of how we can glean some of these data just from routinely acquired clinical information. And one of the things that we've then gone back to do is take some of these early stage kidney cancers So they're depicted here in green, and in gray are all the larger tumors. On the y-axis is the number of subclones. On the x-axis is the size of the primary tumor. So as you can see, the punctuated evolution tumors are ending up at very large size, but they have very limited number of clones, while branched evolution tumors are growing to a large size with a large number of clones. And these early stage RCCs, some of them were showing evidence of budding, but not all of them. And this gives us some hope that by observation of these radiological images, we might be able to assign the likely trajectory of these earlier stage tumors. And we are currently doing this in a larger scale exercise.
Hi, Samra.
Yes.
Yeah, can I ask a question?
Of course, please.
Yeah, can we go back one slide?
Yes.
Just wanted to understand, how do you define subclones here? Is it independent of the clonal growth itself, or is it just like mutational landscape?
Yeah, of course, thank you. So in the way that we've defined clones in all our work to date has been based on the presence of a driver mutation in a particular branch of the tree. So our clones are certainly reflective of selection as opposed to neutral evolution. And I think even the sort of pan-tumor neutral evolution studies have frequently found that the role of selection is very strong in kidney cancer. So we don't tend to have branches that are driverless. So the number of subclones is the number of genetically distinct populations that are characterized by a separate driver event, whether that be a mutation or a copy number alteration.
Okay, thanks. Yeah.
You're welcome. So, of course, we're interested in how the lethal phenotypes emerge. So, again, in what we are helped by in the framework that we use here is that we have finally resolved clonal composition of the primary tumor. Therefore, if we have a paired metastases, we can pinpoint the origin of the clone that founded the metastases in a distant site. And when we did this in our interim analysis, we were able to do this across 1,200 primary tumor samples and 335 metastatic samples, and we individually analyzed 250 clones. And essentially, we asked a very simple question: what is the difference between these clones that we don't find as founders of metastases and those clones that appear to be metastatically competent? And they had the same number of driver mutations, but they invariably had high levels of chromosomal events, and very specifically, we see that they were enriched for loss of 9p and loss of 14q. Now, we're currently developing preclinical models to understand how loss of 9p and 14q affords the metastatic phenotype. I mean, some of the sort of obvious candidates here are pointed out. So JAK2 and CDKN2A on 9p, and HIF1 alpha and 14q, which is a known tumor suppressor gene. But bear in mind that the remaining allele still remains. So this is not necessarily the target of these copy number events. And what's interesting is that we continue to see, especially in polyclonal tumors, so branched tumors, that lack, for example, 14q loss by bulk profiling. We here see that there is emerging evolution of driven by 14q at single cell level. So this might reconcile, another rather unique observation in kidney cancer, which is that when the tumors have already metastasized, in certain situations, removing the primary tumor actually helps the impacts the overall survival of the patient, which is really a precedent in solid tumors. And that suggests that some primary tumors that have this sort of ongoing evolution and a chance to still fixate further metastasis affording events if it's possible to interrupt that process, then perhaps the metastatic burden can be reduced. The other thing that we found was that 9p and 14q also frequently co-occurred. So this is a co-occurrence map. And therefore, we were really pleased when we saw these are collaborators of ours, in fact, at MD Anderson, who developed a mouse model of kidney cancer, which isn't, it doesn't actually have those sort of early canonical events. But it's targeted to the proximal tubule, actually involves a knockout of NF2 and SETD2. But once chromosomal instability was generated in this mouse model, there was very quick fixation of 4q, which is synthetic to human 9p in the mouse, and then subsequently selection of 12q, which is the mouse equivalent of 14q. So, so this suggests that this evolutionary bottleneck is recapitulated across species. And again, we're functionally investigating how these combine. I'm sorry, something's happened with the presentation here, so you really can't see it. But all I wanted to show you here is how the timing of these high-risk events influences the mode of metastatic dissemination and the composition of metastases. So here's a patient who unfortunately ended up having a post-mortem only 6 months after their original diagnosis of a primary tumor. They had widespread metastatic disease, and their primary tumor was dominated by this clone that had 9p and 14q loss, and every single metastasis was seeded by that clone. But the metastatic spread in this patient was very different. So this is a patient that had a much more attenuated disease course. At presentation, they only had a primary tumor. It was very slow dissemination, firstly to a pancreatic metastasis, and then many years later to other metastases. And we see that this is a much more polyclonal tumor. It follows that trajectory of loss of VHL, then PVRM1 is a major clone, then repeat selection of SETD2, and then this minor clone that's characterized by loss of 9p and 14q. And this indeed is the clone that seeded all the metastases, with the exception of pancreatic metastases. And I told you earlier that these endocrine metastases are often very, very indolent. And we were surprised to see that this metastases, although this clone was, if you like, available at the primary tumor site, this metastasis was in fact seeded by the ancestral clone, which has very low levels of proliferation. And when we looked further into other matched primary tumor pancreatic metastases pairs, we could see that this was actually this seemed to be a fairly consistent observation, that pancreatic metastases was seeded by the ancestral clone, is irrespective of what other clones were evolved and fixed in the population. And this essentially means that these particular metastases have very low levels of chromosomal complexity. They often only have VHL and 3p. And it brings into question this assumption that these are dormant metastases that are then subsequently exit dormancy either through angiogenic switch or loss of immune surveillance, and it suggests that possibly they're just very, very slow growing metastases that are only detected very late for that reason. And then just turning back to our framework of matching the genetic composition of clones to their spatial spatial features, and sorry, this is yet again is somehow being shifted to the right. But we specifically asked about the difference between the clones that are found in the tumor center and the clones that are found in the tumor margin. So this is shown in dark brown. And we find that the tumor center appears to be a harsh environment that selects for clones that have increased levels of copy number alterations, increased proliferation, higher grade, high levels of necrosis, and finally, metastasizing clones. So those metastasis competent clones were far more likely to be found in the center of the tumor. We also could see that the the clones that acquired copy number alterations beyond 3p loss, even though they were happening later than mutations, actually drove the largest clonal expansions in the tumor. So this is genomic distance versus spatial distance, and we see that these clones travel very far relative to the ones that only have mutational events. So what's really happening here? Again, we went back to our friend and colleague Zhao and asked him whether he could model what happens in tumors that have necrosis in the center. And the conclusion from his modeling was that necrosis allows for selection of these highly fit clones through creating more space, essentially. Now, the interaction of all these factors and the direction of causality is, of course, completely unclear. So necrosis happens as a result of either acute or chronic hypoxia, and both can happen in kidney cancer. So there can be acute infarction through thrombosis of a vessel that's supplying a population of cells, or there could be chronic environmental hypoxia because of inefficient angiogenesis. And exactly how all these are ordered temporally is something that we're again studying functionally at the moment. So these are just very briefly conclusions from what I've told you so far, and I'll move on quickly to perhaps just tell you a little bit about our current focus. So one of the limitations that we have with studying sporadic cancers is that there will be many unique features to each instance of a sporadic cancer that are determined by some patient factors. And that, again, is why we turn to mathematical models and preclinical models. But we were also really interested in searching out a proper in vivo model, so a patient model, or where this opportunity to rewind tumor evolution might present itself. And this really, the opportunity presents itself through studying of VHL disease. So this is an autosomal dominant disease where patients inherit mutations in VHL, inactivating mutations in VHL, and present with quite constrained clinical phenotype. So they get hundreds of kidney tumors and cysts. They get hemangioblastomas in the brain. They get pancreatic neuroendocrine tumors, and they get adrenal tumors. So, for example, they don't get lung cancer, they don't get colon cancer, they don't get breast cancer. And of note, in sporadic cancers, VHL mutations are really only seen in clear cell cancer. So there's a profound tissue specificity. But what's elegant here is that VHL disease provides a kind of a mirror image of genetic ordering of events that we see in the sporadic setting. So with this germline constraint of VHL mutation, the second event is loss of 3p, while in the sporadic setting, it's loss of 3p first and then a VHL mutation. But what we really focused on is this idea of repeat evolution of multiple clear cell kidney cancers over a lifetime. And this has been a wonderful collaboration that we started over the last 18 months or so with Marston Linehan at NIH. And of course, Marston and his team described VHL mutations in these families some 3 decades ago. And they see many patients with VHL and manage them over lifetime. And I've frequently said during this talk that we don't have time course resolved data in cancer, but actually in this instance we do, because these patients will be monitored with this burden of individual kidney tumors, clonally unrelated tumors in the kidney, until they reach this threshold, which is 3 centimeters. The reason for this is that, Dr. Linehan has described this in detail and published, is that tumors that haven't reached 3 centimeters will not metastasize. So that in itself is a fascinating finding. But for us, what's important is that when Dr. Linehan and his team go in to resect the tumor that has reached that threshold, they will also try and reduce the burden of disease, and therefore they will procure other tumors which might be regressing or stable, or they might present one of these cysts. So we're now getting readout from a variety of clinical phenotypes, and we can decipher their molecular profile. So one thing that this is very recent data, just over the last month or so. One question we're asking is to what extent, when we're studying 10, 30, 50 tumors from the same patient, do somatic events cluster between or within patients? And within this, we even have some families. So there we've fixed the germline variant, but we haven't fixed the environmental influence. And we do certainly see examples where there's non-random convergence of driver events, both somatic copy number and mutation profiles in some patients, but we don't see it yet as a universal rule. So we're trying to understand at the moment whether the tumors that we're studying are sharing the same microenvironment in some instances, or whether they're more separated. Now, the other thing that I mentioned earlier is that the VHL phenotype represents a number of different clinical entities in the kidney. So some of these lesions will be solid tumors from the start. They will grow over time. Some of them will present as cysts. So this is just a monolayer of cells, and there is a fluid-filled cavity within it. Some of these cysts, as Dr. Linehan observes over time, will then become mixed. They'll have a solid component, so this little nodule will appear on the surface and they become solid over time. But actually, some of these cysts will just persist as cysts throughout. So we were very interested to understand what's happening here. And again, these are very early insights. We need to go over a larger number of samples. But the picture that's emerging is that we're seeing loss of 3P as a sort of obligatory event for a solid tumor to emerge, while in cysts we're seeing an enrichment for a second event to be a mutation in VHL. Now, as you've seen throughout the presentation, loss of 3P doesn't only result in inactivation of the second allele of VHL, but actually opens up an evolutionary path because of LOH and those other tumor suppressor genes that are located on 3P. So this is potentially really beautiful correlation between genetics and the phenotype. Just keeping an eye on the time. You can let me know what you'd like me to do. There's some data in melanoma that I wanted to show, and then maybe just very briefly at the end, something about the methods that we're developing to capture evolutionary features in the clinic. We're very fortunate to be part of this national study, post-mortem study in the UK. My colleague Mariam Jamal Hanjani at UCL Cancer Institute initiated this study as the chief investigator nationally. We opened the study at my unit here at the Marsden, and have been enrolling patients both into melanoma and kidney cancer cohorts. And as I've alluded to earlier, we're able to access the types of metastases that we can't access in life. I'll just tell you briefly about our insights into emergence of lethal melanoma from this cohort. This is a cohort of 14 patients that we've been studying. I'm not going to go through detail of the mutational profiles. I really just want to talk about copy number changes today. So the thing that we have been focusing on is whole genome doubling, because there have been conflicting studies in melanoma, some suggesting that whole genome doubling is an obligatory event for progression to metastases, and others suggesting that this is more variable. And what I've circled here, so these individual bars at the top are patients, and then below are the samples, all the samples that we've collected from those patients. They're from a variety of study sites. And what we see is that in some patients, whole genome doubling is indeed absent. So there are other routes via which these patients have progressed. But in some patients here in dark blue, we see universal whole genome doubling, but actually not just a single round of whole genome doubling, but in fact multiple rounds of whole genome doubling, which really took us by surprise. And what's clear is that obviously whole genome doubling per se is not advantageous. It's what it facilitates, and what we see clearly is that the more whole genome doublings you have, the greater the proportion of the genome that's altered by copy number. The timing of whole genome doubling tends to be early in some patients, and it tends to be subclonal in other patients. And the pictures that I'm showing here are of fish experiments that we felt we had to do when we first observed this, because the multiple rounds of whole genome doubling, because we thought that maybe this is some error created through the nature of these samples or our computational pipelines. But in fact, we confirmed multiple rounds of whole genome doubling in these samples. But furthermore, what we see is a lot of cell-to-cell variation in ploidy. So it's not just about fixed aneuploidies in these patients, it's also about ongoing SYN, which I've just listed some of the ways here in which SYN can then feed immune evasion, which is what happens. It happened in all these melanoma patients who received immune checkpoint blockade. And what's really interesting is if we compare end-of-life samples that are diploid with those that are whole genome double, the copy number gain profile looks very similar, but there is extensive copy number losses and deep copy number losses in those tumors that are whole genome doubled. We see that those copy number losses encompass tumor suppressor genes, antigen presentation genes, and occur in regions that are linked to immunotherapy resistance. Now, this is a small cohort of patients, so we can't really make any major insights relative to mechanisms of resistance because we're dealing with small numbers. But because we're dealing with many lesions, we're able to go back and study the individual radiological responses to therapy. So when we leverage all the lesions across patients, we actually have quite a large cohort of lesion-specific responses to look at. And we see that the copy number profiles of non-responders here shown in blue are characterized by amplification in MYC, which has been linked to immune evasion by the work of Jared Evan and others. So just very quickly to conclude here, there are multiple routes to lethality from what we see so far in melanoma, but whole genome doubling really facilitates onset of chromosomal instability and LOH and potentially treatment resistance. And I'll be very quick to allow for questions on this last part. And this is kind of an inevitable theme to come to in the end, because it's clear from the data that we generate that there is a serious problem with undersampling of tumors in the clinic. And it just illustrated it here. That even the sort of work that we do in the research framework, which is multi-regional sampling, really undersamples the tumor as a whole. So especially in these very large tumors, and those are exactly the tumors that need to be understood, because they tend to confer a poorer prognosis and are more likely to be heterogeneous. And this is just this is published data. This is just a representation of this undersampling, both in the research setting and the clinical setting. So what we developed over the last few years is this idea of representative sequencing, where we homogenize leftover surgical tissue. So this is trying to bring us closer to something that a hematologist would have when they take a blood draw. And that they will always recover a mixed and a representative population of cells. So not only are we trying to get a greater representation of the tumor as a whole, so more cells being represented, more populations being represented, but we're really after this mixed population, so that every time you test a sample, you're getting the same phylogenetic tree, or you're getting the same biomarker data, as opposed to taking repeat samples and getting different readouts each time. And I won't go over this because this is published data where we've shown the reproducibility and the new discovery, there's increased discovery. But I just want to show you this case. So this is a large kidney tumor where we did multi-regional biopsies, 60 multi-regional biopsies, to capture these two major clones, one in purple and one in green, and the lethal clone, so the one that formed the metastases being in purple. So contrast that or compare that to the phylogenetic tree that we recovered by sequencing just a single sample, which is this representative sample. And indeed, the cancer cell fractions were very comparable, and the shape of the tree was very comparable. This clone D, which was a lower cancer cell fraction, was not detectable by this method. So it's probably not ideal for very low frequency events. On the basis of this, I'll skip over this because it's published. We've moved into enriching these homogenized samples for tumor cells so that we don't have to do so much redundant sequencing, and we can discover more branches in the tumor, so sort of overcoming that sensitivity issue. And we were really quite surprised that we were skeptical about our ability to sort these fixed cells, but this is really been now validated extensively, and we've published this protocol at the end of last year. We're now running a prospective study across many different cancer types to test the feasibility of this method in the clinic. But what we're most excited by is that we will get this insight into cancer evolution, or the sorts of tumors where we've just not had it. So there are some tumor types that tend to be extremely large, such as sarcomas, that are very underrepresented by single biopsy study, where we're hoping to get at the questions about the pervasive evolutionary trajectories and the true extent of clonal diversification in tumors in these representative samples. I'm just quickly going to show you, this is, for example, a neoadjuvantly treated breast cancer. So this is the profile that you get by doing single sample analysis, as we would do routinely in the clinic. You detect the major clone with founding mutations, but in this enriched homogenized representative sample, we're detecting ESR1, which is being selected under the pressure of treatment with neoadjuvant anti-estrogen therapy. So potentially we'll be able to predict the cause of relapse in such patients. So as you can imagine, huge number of people contribute to these studies, both from patient recruitment and then into doing the actual work in the lab and doing the analytical work. So I just want to thank everyone in the Crick and the Marsden, and a huge number of wonderful collaborators that we're very privileged to work with. Of course, patients and their families and our funders. And I'll just leave you with this, if that's okay.
Well, thank you so much, Samra, for a fantastically interesting talk. I think we've still got some time to squeeze in a few questions. So if anyone wants to ask a question, I suggest you just unmute yourself, and hopefully we'll be able to coordinate that.
Yeah, can I go ahead and ask one right now? So very, very beautiful talk. I really like how you're saying that the tumor is an ecosystem. Basically, you have this ecological profiling of the clones, and also there is a spatial dynamic that controls a lot of things. So I was wondering that when you see metastasized clones, does this kind of ecology or spatial dynamic is retained in different tissues as opposed to the original sample?
Yeah, it's a great question. And the first confession is that we've focused on molecular evolution because that's easier at the moment to resolve. And of course, it's the phenotypes, not genotypes, that are being selected. And a lot of the work that we're doing now is that understanding the features of the tumor microenvironment where these clones are likely to be selected, and then whether those features are translated at distant sites. And that's the work that we're doing in the post-mortem setting at the moment. So I guess your question is whether they recreate the environment in which they were originally, or whether they no longer depend on that. So, for example, if the adaptation to hypoxia in the center of a primary tumor is to select for these karyotypically abnormal clones, and then a acquire pro-migratory phenotype, once they've disseminated, they're probably not going to experience hypoxia to the same level, certainly not in the circulation, and certainly not initially when they form a metastasis. So there's probably some other aspect of adaptation that chromosomal instability and aneuploidy afford that allows them to found metastases at distant sites.
I was even more fundamentally, if you just think about, so you showed some examples where you have initially had a heterogeneous clonal pattern, like attenuated cancers, which might develop a 9p or other, I forget the name. So that kind of mutant, they start metastasize. So do you still see splinter, what was the third mode of evolution which you had? Which was like,
punctuated, yeah.
Yeah, punctured evolution versus the branched evolution. So do you see the same kind of clonal structure, or is it just because it has taken over the whole clonal dynamic that one single
so
mutation?
I think it's really about timing, because if these events are fixed at a time where you already have a very large tumor volume, it's unlikely that they're going to cause a clonal sweep.
Okay.
And the tumors that fix these events very early on, where the intermediate populations are weaker, but not so large in terms of population as well, those populations will be much more vulnerable to just drifting and being taken over by the clonal sweep. Of course, the conditions under which you would fix these events early and you would fix them late is what determines when they happen, as opposed to, you know, they're more likely to occur in the first place. So obviously the mutations are random. It's the selective advantage that will.
Yeah, that makes sense. All right, thanks.
Thank you for the question.
Do we have any more questions? I have a question potentially while people are thinking about theirs. So with the VHL disease patient cohort that you've looked at, and this interesting observation that if the second hit is conferred by a mutation, you sort of see these dead end cysts arising, whereas 3p loss serving as a second VHL hit often leads to a tumor. Is there also biallelic inactivation of the 3p tumor suppressors, or is it just that one hit in the SWE/SNF subunit, for example, is enough to direct that?
Yeah, so they don't necessarily have secondary mutations in 3p genes when they become solid tumors. So it would suggest that there is something else that also occurs just in that combination of VHL and 3p loss. Potentially, you know, tolerance of chromosomal instability, for example. That means that these will solid, you know, they will present initially as solid cancers rather than growing as a monolayer. I
mean, just the naive devil's advocate, the mutations that lead to cysts, they're strictly pathogenic in activation.
They are pathogenic.
Okay. So there's definitely okay.
So we still see them, but it's a good question. But we see them clustered in the usual.
But effectively, you might argue that if you have biallelic inactivation of VHL, simply monoallelic inactivation of one of these other tumor suppressors that is on 3p can determine whether or not the cancer is tumorigenic or not.
Yes, so we don't know whether there are some other haploinsufficient genes on 3p that are contributing to tumor growth in the first scenario, and obviously there might be, again, emphasis on the fact that we're detecting these very large clonal expansions. So certainly there could be other clones that are contributing to these tumors becoming solid that we haven't detected by our methods yet. So these are single sample studies, so we have less resolution.
Yeah, of course.
But they're good. It's good food for thought, definitely. Thank you.
Okay, are there any other final questions before we conclude? Please interrupt now, if not. We'll say thank you very much to Samra and wish you a good night.
Thanks very much. Thanks.